We're going to look at the kingdom fungi. In this, we're going to produce a lot of material to look at because the fungi are probably the most important plant pathogens that we find. They are to plants what viruses and bacteria are to humans. Most of the plant pathogens that you will find are going to be fungi. The fungi are very complex. Basic definition of kingdom fungi. Eukaryotic, heterotrophic, cell wall is made out of chitin, it is not motile in the trophic phase, and it is filamentous. The filaments are called hyphae. We're going to look at all of these different parts. If the mycelium is cenocytic or unicellular, and asexual spores are motile, we have the chytridiomycota. If the asexual spores are non-motile and the mitospores or asexual spores are numerous and found on the inside of sporangia, we are going to classify these in the zygomycota. If the mitospores are individual or in groups in the root system of plants, we're looking at the glomeromycota. Now you can see the word mycota in there. Myco refers to fungi. Mycota refers to phylum of fungi. And so we are going to look at all of these. Within the kingdom fungi, if the mycelium is septate, which means it's going to have cross walls, and we're going to see this. If there are no sexual spores, they are found in the mitosporic fungi. If sexual spores are present, then we classify them based on whether or not the sexual spores are internal, in which case we're talking about the ascomycota, or if the sexual spores are external, we're talking about the basidiomycota. Basic characteristics of fungi. They're eukaryotic, they're heterotrophic, they have a cell wall made out of chitin, they are multicellular, and they lack motility in the trophic phase. They can be single-celled. This is a yeast, and you can see the single cells. These yeast reproduce by budding, where they produce a small daughter cell coming off of the cell. They could be single-celled. This is Taphrina. Taphrina is a yeast-like organism that occurs on plants. They could have hyphae. The plural of hyphae is mycelium. The hyphae are nothing more than individual little strands or filaments. In this particular case, these are cenocytic, which means that they do not have cross walls. These are septate, which means they do have cross walls. If you look at these filaments here, you can see the very distinctive separations across them, which give you the cross walls. We have what we call a thallus, and a thallus is basically a rudimentary undifferentiated body. This is called a fairy ring, and you can see the large area that this actually covers. This is a single organism, and basically it started out growing in the center. Most of its body is underground, and then occasionally it will pop up these mushrooms out of that. It gathers all this nutrition underground to store up all these energies so that it can pop up these mushrooms when the conditions are favorable. Mushrooms, part of a thallus. It's really an undifferentiated type tissue system. This is a thallus. And you can see how this is growing in culture media. What you've got is a lot of filaments in there, and it's an undifferentiated plant body. Let's go back, look at taxonomy. Taxonomy, break it down. Mycelium cenocytic, we're talking zygomycota for the most part. Zygomycota is going to have non-septate hyphae. If the mycelium is septate, which means it's got septate hyphae, and you can see the cross walls in here, we are looking for sexual reproduction. If we have sexual reproduction and the sexual spores are internal, we are in the ascomycota. When we deal with the ascomycota, we will actually look at the way the spores are formed. They're formed within the concept of this diagram. The basidiomycota has the sexual spores external. You can see here how the spores are produced on top, whereas when we looked at the ascomycota, the spores were produced internally. If there's no sexual reproduction, we call it the mitosporic fungi. The mitosporic fungi produce other mechanisms by which they can reproduce, but they are all asexual in nature. When we deal with fungi, we have to understand the concept of hyphae and mycelium. Hyphae are the individual strands. Myceliums are groups of strands. You can see in this particular case, you've got a filamentous type hyphae. This is a filamentous type hyphae, or a mycelium. It's a collective of it because you've got a lot of these filaments on the inside of this particular plate. Other key terms we have to understand. Dikaryon. Normally we talk about diploid phase. In the diploid phase, you get a nucleus that is formed of part of the genetic material or the chromosomes from the one parent and part from the other parent. In the dikaryon, when you get the two gametes fusing together, the nuclei don't fuse. They stay separate. So what you've got is male and a female, or a plus and a minus haploid nucleus in each cell. rather than having a combination of the two. We have what we call a sporangium. A sporangium is a cell or sac inside of which spores are produced. Very important term to understand. We have chitin. Chitin is the carbohydrate that's the main ingredient of the cell wall of fungi and the exoskeleton of insects. They actually think that fungi may be close to insects because we've got a similar type polymer in the outer wall of it. From this point on, we're going to look at different groups of fungi and what the different groups look like and what they do.